a lot. So yes, good afternoon. My presentation is about how we can use. I'm not going to talk so much about results from the AQUA-FAANG project, but I'm more like the principles of how we can use the information from this project to improve breeding and increase genetic gain for sustainability traits in aquaculture. So I'm Sigbjørn Lien. I'm located at the Norwegian University of Life Sciences in Ås in Norway. I'm coordinating the AQUA-FAANG project together with Dan McQueen at Roslin Institute. And we have a very solid, good group of partners in the project, so altogether 24 partners. So a lot of the output from this program will come later on when we are now in the final phase of the project, so ending in October this year. So for aquaculture, I think genomics and breeding has been very successful, and there has been a lot of benefit already gained from genomics and breeding. As Ante touched upon, we see that by using genome-wide association studies, we identify trait variation associated with important traits. There has been ongoing a lot of research that identifies major genes for important diseases, which can then be selected for to improve resistance to diseases. And there is also breeding companies have put genomic selection into action to improve more complex traits, which are not controlled by a few genes, but by multiple genes, more complex genes. So one of the really success stories so I'm working mostly in Atlantic salmon, as many Norwegians, because that's the big industry in Norway. But for Atlantic salmon, the detection of this a locus on chromosome it's explaining 80% of the genetic variation for pancreatic necrosis in Atlantic salmon, IPM. That has been implemented in breeding and probably since 2008, 2009, and it has kind of reduced the number of outbreaks for IPM dramatically. So this has really been a success story in aquaculture. But this is one disease where we find one gene explaining a lot of the variation. But most of the traits, they are quite complex. They are controlled by many genes, and they can't really be improved much by QTL selection. So then we have the alternative way is to do genomic selection, and that approach is very effective when you work among close family members. But it becomes less effective if it's run across more distant relatives, so across generations, so between populations. And another problem with this approach is that if you want to improve diseases by this approach, you have to continuously do challenge testing for the disease, which is very costly and is also very unwanted for animal welfare reasons, because then this is considered as animal testing, and there is ongoing discussion if this will be prohibited, so not allowed to do it in the future. So we need alternatives way of how can we use functional annotation to improve these current breeding approaches. And here maybe we can learn something from many genome-wide association studies, not only in aquaculture species, but in many species, including humans and livestock. And these studies showing us that it's most of the underlying functional variation is not it's not found within genes. It's rather in regulatory sequences of genes that accounts for most of the genetic variation for complex traits. And these are genetic variants outside the genes. They are proximal. They can be in promoters close to the gene. They could be more distant to the gene enhancers or silencers. Also, quite important, I think this is kind of more new knowledge, is that these regulatory variants are often not single point mutations or SNPs, but they are more structural variants, larger deletions, insertion, duplications, and so on. So this is the challenge that we are trying to address with the AQUA-FAANG project. And what we are doing in this project is to try to develop functional annotation maps for 6 different fish species that are the most important in European aquaculture. This is done for turbot, European sea bass, sea bream, Atlantic salmon and rainbow trout, and common carp. We also have a link to the zebrafish community here for understanding the mechanisms. So in this project, we are developing functional annotations map for different tissues in immature, mature, and males and females. We have activities on environmental stages in different stages under the development. And we also have a specific activity of immune activity where we use mimics to study we use standardized stimulation of viral and bacterial stimulation. So the techniques we use in this project is methods to measure, to identify the regulatory sequences, and this is done by lab techniques to study the chromatin accessibility using ATAC-seq. We use methods to study protein and DNA interactions using ChIP-seq for histone marks, and we measure the transcription levels of different the whole genome, all genes in the genome. And after that is done across tissues, across developmental stages with different assays, techniques, we start building a regulatory map using these data. And this model is built up as taking the data from the lab work and identifying elements that is identifying promoter sequences, up regulation, down regulations of promoter sequences, or other assays identifying enhancer activities, so that's elements outside the promoter. And we built something we called regulatory build, describing where are the regulatory regions. And this is done by tissue and by individuals that we study. We use here, so far we have used a program called Chrome HMM to do this, but this is also being set up now by Ensemble, and I'll come back to that later on, and made available for everyone. Okay, so the next step after we have made the functional annotation map is to combine the data to predict causal variation. So in most studies today, we are, this is showing where we are today. We are doing a genome scan, whole genome scan for genetic variation affecting phenotypes of interest. In most cases, we end up with a number of SNPs associating with so peaks associating with the trait of interest. And when we try to find what is the causal variation, it becomes very difficult because there is many SNPs in close linkage disequilibrium to each other, and it's very difficult to say, is it this SNP or is it this SNP, or what is the causal underlying reason for the phenotypic differences. So that's where the functional annotation come in, where we can use information about not only the genes, where the genes are located, but also where are the regulatory regions controlling the expression of the genes. So we combine that information with a SNP variation, with regulatory regions and expression of the genes to try to find the causal variation underlying the different traits. And just to complicate a bit, I also going to touch a bit about what is now being developed in many species, including the fish genomes. So people are now also expanding from one reference genome, like we have mostly done in the future, to something called pangenomes, where we sequence not one individual but multiple individuals from the same species. In human, this is very fast evolving, and people are collaborating to make a complete reference of human genome diversity, and this is also happening in other species, including the fish species. And in humans, they have taken this as part to sequence complete chromosomes from telomere to telomere. This is important in order to characterize and map the full genomic diversity within species. It also provides, when you do this, you get some very important information about not only SNP information, so the point mutations in the genome, but you also get the structural variants because you do this analysis with long-read sequencing, and you are able to map out and characterize structural variants. And this is just to say that this is quite important. So this is a paper from tomatoes where it's published in Nature in last year. And you see here, when you take this whole multiple genome approach, with the pangenome approach, and you study and you take that whole genome, including SNPs and SV information into account, you are able to describe more of the heritability of different traits. So here, in tomato, on average, for all the phenotypes they have on tomatoes, they increase the heritability of the traits from 0.33 to 0.41. So this is considerable. So just by describing more of the genetic variation in the genome. So what we are doing now in Atlantic salmon, where we mostly work, is that now we have expanded from one reference, which is the current system, to currently we are working with 11 genomes, and we use long-read sequencing from these genomes to identify structural variants. And structural variants, that's larger deletions, insertions, duplications, and translocations, inversions in the genome. They are by far more difficult to find and characterize and genotype, but there is a lot of it. So in this, in Atlantic salmon, we identify more than a million structural variants. They overlap a lot of genes, more than 2,700 genes, and they capture a lot of DNA in the genome. So 368 megabases of sequence, that's more than three chromosomes in length. And they impact by far more DNA in the genome than SNPs, at least 10 times, probably in salmon more than 30 times the amount of sequence if you compare to with SNPs. So this is something that this information will grow, and there will be more and more information about this, and this information has to be taken into account also when we are studying functional variation. So now I will just take a quite random example. I took just one paper from Nofima, our neighbors here at Ås. It's a paper about genetic variants associated with omega-3 fatty acid composition in salmon fillet. And in this study, it's a GWAS study identifying a number of loci in the genome affecting the synthesis of omega-3 in Atlantic salmon. And one of these peaks identified a gene called a peak close to ELOV2, which is a known candidate for conversion of DPA to DHA. So I took this as an illustration. I took this gene and I displayed it. So that I took the SNPs from the 220K SNP array in Atlantic salmon, so 220,000 SNPs. And I asked the question, how many of these SNPs on this array will overlap coding sequence in this fatty acid elongase 2 gene? And it's none of them overlap coding sequence. Then I asked the same question with the SVs that we have identified, and you see here that these SVs, they affect much more sequence than the SNPs. So we have 8 SNPs here, but we have large insertion deletions with up to 340 base pairs, so much more affecting much more of the variation. But none of the SVs affect the coding sequence. So if this gene is a candidate, the kind of the best candidate gene for this phenotype, how can it be that it needs to be something else? And that's where the functional annotation came into maybe important. So then I took the promoter, so that from the functional annotation in the AQUA-FAANG project, I took those elements that is identifying the promoter activities and the enhancer activities close to this gene. And then you see that so 2 SNPs here, for example, are overlapping promoter activities. So they may regulate, may be important for affecting the transcription level of the gene. There are also deletions overlapping this, affecting much more DNA sequence. But there are also other regions more distant, so that could affect enhancer activity of the gene. So this information can then for the breeding companies, they can use this information from AQUA-FAANG and say that when we will develop new arrays, or we will develop genotyping arrays based on SNPs or SVs, we can select those that are likely or possibly functional instead of just taking a random SNP or a random SV in the genome. So that's I think the important message here is to say that the functional annotation maps, they reveal regions in the genome that are affected. We can use this information if we are able to identify functional variants, and we include them in genotyping arrays. They will work much better for when we use them in selection, across generations, and in different populations, because they are functional. And the way this can be done is that breeding companies, many of the larger breeding companies, they have SNP arrays already developed, but they can replace some of the SNPs they have on their arrays with new ones and with functional variants, potentially. For other species, it's maybe more relevant just to develop new genotyping products to do this for SNPs. But also, I think in the future, there is a demand for new technologies, more cost-effective technologies to really genotype the structural variants, because they are not easily genotyped with SNP arrays. So this is a challenge also for the future. And then, of course, like Antti talked quite a lot of, we need to get this information into programs to predict the genomic predictions to improve. So make this into breeding programs. Okay, so and these tools that we develop in the AQUA-FAANG project, they are currently in development, but they will soon be available through Ensemble. They don't in Ensemble, they don't do Chrome HMM like I showed, but they have a different approach called, and they build regulatory bills, but basically doing the same thing, identifying the regulatory elements in the genomes. There are also tools here to predict the functionality on SNPs and also to some extent structural variants available online that the users can use to predict the functionality of variation. And this is my last slide. It's just to acknowledge all the good team in AQUA-FAANG that is kind of collaborating on developing this new tools. Okay, thank you.